Welcome to this video tutorial exploring how you will analyze your own single-cell RNA sequencing dataset using the software AltAnalyze. Before you begin, you should have either yours or a publicly available 10x Genomics RNA-seq dataset file. We prefer these H5 files. These are produced from the Cell Ranger software. These are sparse matrix datasets. They're usually fairly small. In this case, this file is 16 megabytes. Before you begin, you'll need to have AltAnalyze installed. So download that program if you haven't already installed it from the AltAnalyze website. Once you've extracted it to your computer, either typically on a Mac or a PC, you're going to open the program folder. You're going to find the green AltAnalyze icon on a Mac or the white AltAnalyze icon. Sometimes the green AltAnalyze icon doesn't work depending on your system. On a PC, you're going to double click on the altanalyze.exe. When you start the software, you're first going to be prompted to begin analysis, to continue, and then to download a species database if you have not done so before. This requires a connection to the internet. You first select your species of interest, in this case Homo sapiens, and then continue to download your database. Once this is complete, you can proceed to the next step, in which we'll import that H5 file and process it in the software to generate unsupervised clustering results, annotate those clustering results, and visualize the output of the program in various ways. As soon as the database has completed installation, you'll be ready to analyze your 10x Genomics dataset. You'll be presented first with this main menu, in which you'll see the species that you downloaded, and this option for raw sequence or process dataset. Here you're going to actually select this option and select 10x Genomics aligned. This indicates a sparse matrix file such as the one you see here. Selecting continue, you're going to select process Chromium matrix. We're going to give the dataset name. In this case, this is a lung dataset, a human lung dataset. We're going to select the file, actually in this case, that is an H5 file. Now, if we had wanted to here, we could have selected a folder, a directory of multiple H5 files or .mtx files to integrate. We should also select a folder in which we're going to save these results to. In this case, we're going to save them to the same folder that the H5 file is in. Selecting continue. There's a number of program defaults you can select. In this case, for an unsupervised analysis, you really don't need to worry about these other options, so you can just select continue. Again, there's all these pathway analysis options. For the unsupervised analyses, these don't really apply. So again, select continue. And when you get to this window, you have a choice of either assigning each cell, which we're not going to do, to a predefined group, or running a de novo cluster prediction using the software ICGS, specifically ICGS version 2. Selecting continue, you'll be presented with these defaults. Typically, these defaults will be fine for you to apply. You can vary the different clustering methods, the number of cells to downsample. Cells are downsampled intelligently. They're not randomly downsampled. They're effectively pre-clustered before they're sampled. And you can select any other options you want. When you're done, you select Run Analysis. The program will proceed to process those files, to generate any intermediate files, identify initial clusters, and then identify final clusters using a method called non-negative matrix factorization. In this case, the dataset has 6,800 cells, and so by default, it's going to downsample it to 2,500 cells. You can set your own number of cells to downsample to, or not downsample at any. In this case, you would just set downsample to 7,000, for example. This may run from anywhere from 5 minutes to 20 minutes to an hour, depending on the number of H5 files and the number of cells that you have in your dataset. So be patient. You may want to walk away, get some coffee, hang out, and we'll be right back. When the analysis is complete, you'll be presented with the following dialog box that says Predicted Sample Groups Saved. You'll also note that there are a number of new windows that have been opened up here on the computer that say Go Elite Results. We'll go into that a little bit later. For now, you can close these windows and select OK for the Predicted Save Groups dialog. You will be presented with a series of graphical icons that display the different clustering steps involved in ICGS2 to identify final clusters, in this case cluster 7 here. This is a low resolution image, so you may not want to pay too much attention to it. And this is just because a PNG is, or I believe a JPEG, or actually it is a PNG, is shown in this case, which shows a graphic summary. This looks kind of ugly. However, if you close this summary and you quit the software, you can now go to this ICGS NMF folder on your computer that's now in the folder which you've output your results to. If we open up the Marker Finder Heat Map All, you'll obtain a high resolution version of that image and you'll see it'll be much more clear. If we zoom in here, we'll see a few different things. First, you'll see a number of clusters displayed at the top. These clusters represent NMF-based clusters, so don't worry about the fact that they're not ordered in a linear manner. You will see that the clusters have been organized based on their similarity to each other. So in this case, you have a number of clusters with the term monocyte, monocyte, macrophage, monocyte, DC, macrophage, macrophage, that are all very similar to each other transcriptionally and are all right next to each other. This is because the program is reordering the final clusters based on their similarity to each other, based on hierarchical or HOPAC clustering. Similar down here, you see other subsets of T cells and NK cells and B cells that are most similar to each other. These enrichment profiles on the left are from a gene set enrichment analysis from the software GoElite. The names here are derived from gene sets from various different studies which actually have the referenced citation right next to them. And it says whether they were derived from the top 100 genes from either ICGS2, from a different database, or from going back to the original publication. The cluster number that associates with the cluster on the top is indicated here in parentheses. There's also the top marker gene associated with each of these clusters shown on the right. In many cases, you may recognize some of these genes as well-defined marker genes of those populations. For example, MZB1 for plasma cells, CD3D for T cells. If we go back to the folder, well, we can see that there are a number of other outputs here that we would want to explore in more detail. These are the primary outputs, so we're going to explore these for the next few minutes. First, the first file I opened is this Marker Finder Heat Map All. This contains all over 6,000 cells or so that were originally input to the program, even though we did the downsampling, or the program did the downsampling. The Marker Finder Heat Map All sampled actually shows you the 2,500 sampled cells that were actually analyzed. And this PDF will look almost identical to the one we saw, except these are now the downsampled cells versus all cells. And you should see, even in most cases, very rare cell populations that are present here that you may not have identified through random downsampling, here only through this PageRank-based sampling algorithm. Again, you'll see the same genes, the same enriched terms, and the same clusters on top. However, all cells have now been classified into these different populations. If we look at this Marker Finder All UMAP, you'll see a UMAP which displays all the clusters we saw in the heat map with the number displayed over them and the color corresponding to that cluster on the right. So you can simply go and see which clusters correspond to those gene expression defined clusters and those marker genes. However, there's another file here called Marker Finder Heat Map All UMAP Cell Type. If you select that, you'll actually get a projected label on top of these clusters, which suggests what the likely cell types are in these different subsets. For example, we see AT1 and AT2 cells, which are classical epithelial subtypes. Another epithelial subtype here called ciliated. You'll see an immune subtype predicted here, basophil, different fibroblast and smooth muscle populations, different monocyte, macrophage, and dendritic cell predicted populations, plasma cells, B cells, basal cells, and predicted NK and effector memory cells. Now, these are not necessarily the correct labels for all these cell types. These are predictions from the software based on a gene set enrichment prediction, which is looking at, in this case, hundreds of different studies to predict what the likely cell type names are. To really identify which cell types these likely are, you may want to go back and explore the individual marker genes for each one of these cell populations. In this case, we look at the marker genes all file. We open that up in a program like Excel. It is a tab-delimited text file. You can delete this column here. And here we can look at the marker specificity in each one of these cell populations. Here I tend to like to look at either the top marker genes, or we can look at a certain cutoff here. So if we look at greater than 0.3, and this is the Pearson correlation cutoff associated with the program marker finder within AltAnalyze, this basically indicates the specificity of each gene for each cluster, with a value closer to 1 indicating a high degree of specificity. In the case of cluster 1, GZMH is the top specific marker, followed by CCL5, CD3G, KLRG1, and LCK. Several of these are T cell NK markers, and if you wanted to expand this list, we could simply decrease this filter here and take these genes and input them for gene set enrichment to another program such as TopCell, which we're not going to do right here. But again, that is an exercise the end user is recommended to perform to validate the identity of these clusters, or just look at these markers in the literature. These exist for every single cluster you can see here, and you can just scroll down to whichever cluster of interest. They're displayed by cluster number rather than the predicted cluster name that we saw before. Here again, we see a fibroblast-like cluster based on the marker genes that we're seeing expressed. There are also a number of other files here which you can use to perform a number of other analyses. Here we see the UMAP coordinates. This is the XY coordinates in the cell order that are shown in the heat map for the UMAPs X and Y. We see this final groups file. This is displaying the cell barcode and final cluster groups. You'll see two cluster columns. These are the actual cluster labels that are actually displayed in the final heat map. This is the order of those labels. So if we go to the very bottom, we can see there are 23 final clusters, showing that the last is this cluster 10 label. There's a similar file which shows the exact same results as this file, but now instead of displaying just the cluster number here, you actually see the cluster label that's predicted from the software. So cluster 2 is labeled as adventitial fibroblast. Those are the primary results from the software. You can now manipulate these results further in AltAnalyze to perform downstream analyses depending on the types of data sets you have. This includes going back into the software and actually looking at regenerating a UMAP with different coordinates, or it contains, for example, if this was run with multiple samples, we could go back and visualize where those different original samples show up in the UMAP plot by coloring them differently. Again, in the dimensionality reduction option under accessory analyses, and this has been covered in other videos for AltAnalyze. To see how the results that we obtain with software ICGS match up to those from another program, we can load the same H5 file into an online website that performs supervised classification of single-cell datasets against previously established references. One such tool is the tool Azimuth, which we're now going to go to. So we open up a web browser, open a new window. In this case, we're going to go to the lungmap.net website, which hosts its own version of Azimuth with a lung reference. So by selecting the LungMap Azimuth link, it will open this rShiny browser instance. And we can browse here to basically identify our own H5 file, which we loaded here, which again happens to be a lung data set, and we can compare to this other human lung reference. If this was another reference that was not appropriate for this lung data set, then this analysis may not be appropriate. Once it's been uploaded, you can map these cells to the reference. We may want to set some minimal quality control thresholds here. In this case, just to get results for all cells, we're going to say map cells to reference. And we're going to map these 6,800 cells to the Azimuth reference, which will be a very quick analysis because this is a supervised as opposed to an unsupervised analysis. The potential advantage of a supervised analysis is that there are prior annotations that have been carefully considered and curated from previous studies, which you can see how those match up to your unsupervised results. If your data sets contain other cell populations that are not in the supervised reference, that may be a problem for classification with a tool like Azimuth, and a potential advantage for an unsupervised approach, such as ICGS. Once the results are generated, you have these new options on the left, including cell plots. And so we can see for this data set, as shown here on the bottom, this has been classified within the same UMAP embedding schema as the reference data set shown on the top. And you can see the frequency of your different cells here and in this plot below, which shows the cell frequencies for each one of the predicted cell populations. You can look at specific genes similar to those we were looking at before. In this case, we see a T cell marker, and we can see this marker here. We can select other markers within this plot, which will be automatically displayed here and in this violin plot down here. You can also select markers similar as we saw in the software ICGS results, for example, this AT1 cell population, to see what the top AT1 cell markers are. In this case, we can look at Azure, double click on it, and the results will show here to show that it is an AT1 specific marker, and we can see AT1 markers in this plot. You can download these results importantly, and people will want to download the cell association results where possible, so you can compare those results to the ones that you've generated in ICGS or wherever else. That concludes this tutorial. We hope you have a wonderful day.